is school next door.
Cool.
Yeah, we're coming down the 7th.
You try again.
No.
Yeah?
Yeah.
Is that so all presenting ready to go?
Yeah.
Yeah, shall I go for it, yeah?
Yeah.
Okay.
Yeah, otherwise we're going to rush that.
Yeah, well, I'll... Oh,
Yeah.
Okay.
got till 2 o'clock, yeah? I'm pretty sure it's 2 o'clock.
Yeah.
Yeah, okay.
Yes, that's good.
Oh, yes.
What's that? Yeah. No, why not? Yeah. What does it look like online?
Like editing mode.
Okay.
This happens for us.
Stop sharing, and then I'll just share the screen.
Okay.
As opposed to share the screen instead.
How's that?
Yeah, perfect.
Okay.
So that's fine, yeah?
Yeah.
And then just use this to go
Yeah.
across here. Okay, fine. I'll try and be to the point then, for once. So I'm Ben Carpenter. I'm a adolescent and adult transplanter at UCLH, and have been looking after patients going through allogeneic bone marrow transplant, but also looked after patients on the Bluebird gene therapy studies from a thalassemia point of view, and also we've had some patients enrolled on the recent the CRISPR gene editing studies as well. So what I want to do, hopefully to cover the basis for something for everybody, is to go through the basic principles of cellular therapies, how we go about doing them, and a little bit of a summary of the results, the outcomes, and so hopefully summarise all of these different areas and give you the information you want. Super happy if people want to ask questions as we go along. I'm going to try and be to the point because I've got half an hour now, but what I want is you to have an understanding and get the information you want. So if there's something that I've lapsed into acronyms, etc., from the transplant world, then please pick up your hand and get involved. So having said that, then press the wrong button straight away. There we go. Hopefully that's advanced. And so this is one of the targets. We want hemoglobin A, or as we'll come on to later, in some cases fetal hemoglobin production, erythropoiesis, re-established to overcome the phenotype of both transfusion-dependent thalassemia and also to ameliorate sickle cell disease. And sickle cell thalassemia as a target, any problems, any deficiencies, bone marrowopathies, where the pathology comes from the hematopoietic stem cell compartment, then that's an attractive target for cellular therapies, because we can mobilise hematopoietic stem cells. We can either get them from a healthy donor and replace them with allogeneic bone marrow transplant, or we can take a patient's own hematopoietic stem cells and we can add in a functional gene, or potentially we can correct or alter the gene expression and the hemoglobin production in those cells, so when they get given back to the patient, you can reverse their phenotype, hopefully. And so what I wanted to go into first is how do we get hold of hematopoietic stem cells, what do we need to think about when we're getting donor cells, and also what do we need to do, how do we go about getting patients' own cells for the purposes of gene therapy and gene editing. So you may well be familiar with this, but this is the human MHC system, or HLA, or your tissue typing. And when we talk about donors, we historically have talked about 10 out of 10 donors, or now 12 out of 12 donors. And what we mean is that they're matched from the point of view of their tissue typing, their HLA system. And these are the proteins, these are the genes, and therefore the proteins that we look at. And so we look at the 3 class A and class 1 and 3 class 2 different genes, and you have 2 copies of each, which gives you the 12 different possibilities. And if you match all of them, then you're considered a complete match. And that can be a matched sibling donor, or we have national and international donor registries, and we can go and look for donors from that point of view. Because of the ethnicity of most donor registries, it's often quite difficult to find donors for our patients, and so for haemoglobinopathies, particularly matched unrelated donors sometimes have been overlooked, and I think that's definitely the case in sickle cell disease, but perhaps we'll come back to that later. But when we're talking about tissue typing, that's what we're talking about, that's what we're looking for. And just to remind you of the genetics for the inheritance, because when we say there's a 1 in 4 chance of you having a fully matched brother or sister, then people say, well, I've got 4 brothers and sisters, so therefore it's a shoe-in. Obviously, these are all independent events, but there's a 1 in 4 chance of you being a complete match. So we've got our patient lady in the left-hand corner here, and there's the brother on the far right-hand side who's inherited the shared haplotypes, so that's half of the genes they've got the same half from their mother and the same half from the father and the mother, and so that means that they're going to be a full tissue type match. There's going to be a 1 in 4 chance that you don't share either haplotype, that you've not got any of the genes of the HLA system from either parent, and then there's going to be 50% chance that you share half. So there's a 50% chance you're going to have a haplo or half matched sibling donor, and a 1 in 4, 25% chance that you're going to have a fully matched sibling donor. So it's just something to bear in mind. And then how do we go about getting the cells? So we do apheresis, which I suspect you're all very much familiar with. We can do this, one, to mobilize healthy donor stem cells. So if a donor comes in, a brother or sister comes in, then we can give GCSF injections, mobilize their hematopoietic stem cells into the peripheral blood, and then we can apheresis them off. But likewise, if we want to get hold of a patient's own hematopoietic stem cells, we can give them, in the case of thalassemia, GCSF and also a drug called plerixafor, which helps also mobilize hematopoietic stem cells into the peripheral blood, and then apheresis them off. For sickle, we've avoided GCSF because of the concern about inducing a sickle crisis, and use plerixafor alone. But with that, you can get pretty good mobilization of stem cells into the peripheral blood and then apheresis them off. Then the other approach, and this is sort of much more of the historical, perhaps more definitely from a paediatric point of view, they're used to getting bone marrow as the source for their donor bone marrow transplants, and that's done by the donor having a short general anaesthetic, being laid prone, and then nothing more high-tech than aspirating the bone marrow from the posterior iliac crest. And we normally take about a litre and a half of bone marrow, and that needs a varying degree of processing depending on the blood mismatches and not. And so, a litre and a half sometimes.
Yes.
Yeah. And so, and you do that with a 20 ml syringe, so it takes a while, and you build up your muscles with it. And so for the younger patients, we tend to take a little bit less, but we try and take a litre and a half. And also, if it's an adult donating for a small child, then we don't take a litre and a half, because then you have to process volume, deplete that. But those are the two sources, that's the two ways of doing it. In the very early gene therapy studies, they did look to harvest bone marrow from patients as well, but really with plerixafor, if you're getting someone's own hematopoietic stem cells, then using plerixafor and using apheresis, peripherally harvested, is the preferred way. And then, no matter what approach you're taking, the transplant procedure, the cellular therapies, looks a little bit like this. You've procured your either donor stem cells, or you've got your corrected patient stem cells in your bag, and they can be cryopreserved, or they can be fresh if they're incoming donor stem cells. And then the patients need some sort of conditioning, and it's a common sort of understanding that gene therapy, gene editing doesn't need very much in the way of conditioning. In actual fact, those patients get really very intensely conditioned, and what that means is preparatory chemotherapy to wipe out their own bone marrow, suppress them from an immunological point of view, and we tend to use busulfan, which is quite a toxic chemotherapy, and is one of the sort of important elements of thinking about gene therapy and gene editing. Historically, younger children for donor bone marrow transplant would have myeloablative, or what we call them full intensity transplants, which is really quite intense treatment. But over the last 10-20 years, we've looked to try and dial down the conditioning that people get when they're having allogeneic bone marrow transplants, unless we really think they need it for their therapy, and doing reduced intensity transplants, reduced toxicity transplants, where you're dialing down the amount of chemotherapy that they get in advance. But there's a balance between that the less you deplete the current bone marrow, the more potential residual competition you've got for the incoming cells, and also if they're donor incoming cells, you might have an intact immune response that's trying to reject those cells. Whereas with myeloablative, you're sort of trying to clear the playing field. If they're donor cells, then you'll need some form of immunosuppressant, which is normally started the day before they get their cells, and that's to keep the brakes on the new bone marrow coming in and the new immune system, and that's going to recapitulate and make sure that that immune system isn't reconstituting too quickly, and I'll come on to the reason for that, why. But it's in the months afterwards that a donor bone marrow transplant, the immune system starts to get up to strength as you take that immunosuppression off, and hopefully in time, by 6 months, a year, people will be off immunosuppression entirely. But it's an important time to, one, monitor the stability of the graft, and also to make sure that people aren't running into immune complications of the donor bone marrow transplant. And that brings me on to some of the downsides. So, there we go. With all of these approaches, the preparatory chemotherapy and or radiotherapy is going to make you at higher risk of infections. If they're your own cells coming back, your immune system's going to recover quicker than if it's donor bone marrow transplant, where the immune system takes longer to reconstitute. You're going to be at risk of organ dysfunction because of that conditioning treatment you've needed and develop toxicities from the chemo. An important thing is that people worry about fertility. Fertility is often affected by the conditioning treatment, and so we need to think about fertility preserving options beforehand, and what's possible to give patients a backup option. But depending on the intensity of the treatment you give, that conditioning treatment, sometimes people do retain some of their fertility afterwards, but we always like a backup plan, okay? There's then the risk of secondary malignancies. We know in some of these conditions there's a higher risk of, for example, in sickle cell myeloid malignancies, but if you get given chemotherapy, that is potentially going to increase that risk. It's not a massive increase, but it's definitely there, and it's something that we should acknowledge and something we'll come back to later. But then the last point on that slide is graft versus host disease, and that is relevant when you're getting donor bone marrow and a new immune system. The new immune system recognises immunological differences between the body that it's come from and now finds itself in, and can start attacking healthy tissue, causing rashes, GI upset with colitis, liver dysfunction, and that's one of the things that we really worry about, and that's why you're on immunosuppressants when you have these types of transplants, donor bone marrow transplants, to try and prevent that happening. And when there is tolerance between the new immune system and the patient, then we can ease off those brakes and let them immune reconstitute fully. So there's differences when we're talking about malignant indications and non-malignant indications, particularly for donor or allogeneic bone marrow transplant here. In malignancy, sometimes more is more. If we give them myeloablative treatment, you're wiping out that leukemia, hopefully, whereas we want as lower amounts of treatment to get the graft in if we are doing a transplant for thalassemia or sickle cell disease. We want a new, nice, fiery immune system in malignancy, often to deliver a graft versus leukemia or graft versus disease effect, where that new immune system comes in and helps detect any last malignant cells and wipes them out. That's less important if you're doing the transplant for non-malignant indications. And with that, we like patients to be fully engrafted, have 100% of their cells from their donor when we look in their blood afterwards, whereas that may be or may not be so important from a non-malignant point of view. And we can talk about that at length, but perhaps if we've got time, we'll come back to that. So with the malignant transplants, if you get a bit of graft versus host disease as a sort of byproduct of that, then well, that's deemed acceptable to a certain level. Whereas if you don't need that effect, then giving people graft versus host disease, particularly long-term chronic graft versus host disease from a transplant, or you're potentially swapping one problem for another, which is very much undesirable. And that's why these transplants often have plenty of GVHD prophylaxis, lots of drugs to try and prevent that from happening. So that's some of the considerations about allogeneic or donor bone marrow transplant as a whole. I wanted to just come and touch on the other approaches that are available. So the first of which is gene therapy, and the main option from that point of view is Bluebird Biotech's offering of lentiglobin, which is using a lentivirus an HIV-related virus that is stripped out of all of the genes it needs to replicate, but is able to deliver a new copy of the beta-globin gene into the stem cells. So you mobilise the stem cells from the patient, and then you use this viral vector to introduce a copy of fully functional beta-globin gene that can reinitiate synthesis of that protein, but importantly it's got an anti-sickling mutation in it as well. So when it tries to sickle, it resists that. And whereas for the thalassemia patients, you're obviously starting beta-globin production again by introducing it. And so that again needs cells mobilised, that you need to transduce those cells with the viral vector and make sure that you've done that efficiently, that you've corrected all of them. They then have a myeloablative full-intensity busulfan transplant, but then get their own cells back afterwards. Okay. The next approach is a very clever approach, and I can say that because I didn't think of it, of trying to allow expression of fetal hemoglobin again. So we obviously turn off our gamma chains when we're born, and we stop making fetal hemoglobin as we come into our first few months of life. Well, in the approach of Vertex CRISPR gene editing, what they do is they target the BCL11A, which is a suppressor. It suppresses the production of fetal hemoglobin, and by knocking that out, by disrupting that without using a viral approach, it then allows those cells, once they're given back to a patient, to start in the erythroid, in the red cell lineage, making fetal hemoglobin again. So it's a super clever way of, again, addressing transfusion-dependent thalassemia, but also it means that those with sickle start producing fetal hemoglobin again, and I'll show you some of the results of those studies, and it looks very encouraging. Again, you take the cells, you do the correction, they have a myeloablative busulfan transplant, and they have the corrected cells back, and that's a really important thing to bear in mind. I've got my Duracell battery there. Is the way they get the CRISPR-Cas9 system that does that correction, that knocks down the BCL11A, is by putting electrical current across the cells. It then makes them porous so those molecules can get in. They have a guide so that they are disrupting exactly the right bit, and hopefully it's targeted to just that area, and it's only within the erythroid cells, so the erythroid cells start making fetal hemoglobin again when they're given back and the immune system and the marrow is reconstituted. So, having those sort of three headings of allogeneic donor bone marrow transplant, gene therapy in the form of lentiglobin, the Bluebird Biotech approach, and also the exocell Casgevy or CRISPR gene editing approach. I'm just going to go through the results for, first of all, thalassemia in summary, and then sickle. You're all very well aware of the need and the complications of transfusion-dependent thalassemia. Why I put this up is just to remind me that when we take these patients into these therapies, we need to really think about what organ dysfunction could you expect, and we need to look at those areas in significant detail. So we're worried about heart, we're worried about liver, iron loading. We know that things like veno-occlusive disease is associated with agents like busulfan. So if you've got a very heavily iron-laden liver, or there's been iron loading previously, is there any element of fibrosis? Is there a underlying reason why that liver is really going to deal with busulfan, which isn't particularly liver-friendly? And so thinking, are they fit enough? But think about lung function, fertility side of things, and making sure that patients are fit and are able to undergo these procedures. So how do we do? Well, if we go with the perhaps more classical approach of a matched sibling donor who has a patient who has a myeloablative transplant earlier on in life, and this long-term follow-up is from patients with a median age of 12, so down to very young, up to I think 36, the oldest patient is, but they followed them up into the longer term, and really one of the key things we want to know, well, does it work? Are they making hemoglobin? Yes. Are they transfusion independent? But also, is quality of life improved? Or are we swapping one problem for another? And what this study showed is that those patients' quality of life indices were returning to that, so healthy matched patients without disease, which I think is a really important question for us to ask ourselves as transplant doctors. We do the transplant, it works, but then what happens 5, 10, 20 years down the line? But there's a positive signal from patients actually having had transplants quite a long time ago in that sort of more intensive approach. But what this study goes on to show us is that if you have a transplant early on, particularly the more intense transplants, you do pretty well. You handle them, and your probability of being alive and happy several years out post that transplant is pretty good if you're younger. But actually, as you start to move into your teenage years, people do less well. Now, that could be because perhaps the reason for that patient having a transplant was more pressing, that why did they wait until that age? But we do see this across a number of different diseases, and although I don't consider teenagers to be old, unfortunately, as you start to get into your teenage years, probably the fact that you are, may have been iron loaded, even if you've been collated well, are you starting to develop end organ damage that is making you at higher risk of going into a transplant? And so we know that once you're getting into the sort of 16 to 25 year bracket, then unfortunately those patients are doing less well. What did come out from this study was, though, that if you have a matched sibling donor or you have a matched unrelated donor, you did pretty well. Whereas once you start to have what we call a mismatch, so maybe a 9 out of 10 donor, that they've got one of those proteins that's mismatched, you start doing less well. And so that's less attractive. And also the idea of haploidentical transplants, which we'll come back to later, or half-matched family donors, that's another question that's outstanding. Where's the role for these transplants? But if you want curative therapy and you've got a matched sibling or you've got a matched unrelated donor transplant, then that is something, if you've got transfusion-dependent thalassemia, that we'd think about and say the outcomes are looking pretty positive. I can come back to that later in terms of how we would do it if we have enough time. But moving on to the Bluebird approach, and there was lots of excitement in terms of, the first sort of gene therapy that was being approved for use in patients. Unfortunately, lentiglobin's not available in Europe because it's astronomically expensive, and it's only available in North America. But just to sort of go through some of the initial published data on this, they had 2 cohorts of patients of non-beta naught, so patients who made some beta globin, and then a follow-up study looking at those that who were beta naught phenotype. In the initial study of these, then I think, trying to remember all the numbers here, but just around 30 patients. Initially, as I said, they looked at those with a non-beta naught phenotype, but younger patients, those 12 years and above, were eligible to be enrolled and up to the age of 35. And they had a good spread, some teenagers, some older patients. But in terms of as an approach, it actually looked pretty successful. And so what you're seeing here is in blue, unsupported hemoglobin levels out post the procedure, so good levels of above 10, and in the red line they've got a marker for tracking the hemoglobin that's being produced from the transgene, the gene that's been introduced into their hematopoietic stem cells, and then their bone marrow started to produce. And so really decent levels of new hemoglobin from the transgene working. There are a couple of patients at the bottom that obviously had poor engraftment and weren't producing good levels of hemoglobin. And the patients tolerated the procedure from a transplant doctor point of view pretty well. There was some VOD, veno-occlusive disease, or liver toxicity, which we would probably expect, unfortunately. But the majority of the patients got through. They are intensive transplants. When we've looked after these patients, we've seen decompensated heart problems, we have seen VOD, and so it is an intense thing. But the patients that have gone through our site as a study have all said that they are pleased that they went through it, but it is a tough few months, and that's something that I think has to be underlined. But coming back to this cohort, 20 out of the 22 that were they could evaluate at the time of this publication were transfusion independent, and you can see the swimmer plots of transfusion independent in dark blue there. Two of them had transfusion dependence reduction, but the majority became transfusion independent. And so this is just coming back to the CRISPR approach, and this is something that you will have heard in the press has just been approved in terms of by the MHRA for use in this country, and in the middle of next month it's going to NICE, so we'll see what they have to say in terms of its availability. But it's using that approach of disrupting BCL11A and allowing for fetal hemoglobin to be re-expressed, and you can see just a little outline of how they do it. The cells are mobilized by apheresis using CRISPR and GCSF. They then get edited in the lab. Once you've quality controlled that product, you know you've got enough, you know they're corrected enough, they get myeloablative busulfan, and then you reinfuse that product, and then you follow them up. And so this is some of the data from ASH. Again, they use the same cutoffs. You could go from 12 upwards. There is a younger study for both Sickle and TDD that is now currently open. So they're looking at the same question in younger patients, but it was 12 and above. And they have published results for a cohort of 52 patients. And then there's a primary efficacy set that they've looked at in more detail. Those are those that have been able to meet the primary endpoint, which is 12 months being transfusion independent. So obviously, if you've not had 12 months follow-up yet, you weren't included in that. And so they had 35, which I think is pretty good numbers. Within this study, they went from the get-go, they allowed beta naught patients to be enrolled. And I don't think I'm successfully. Most patients only needed one collection, so in one apheresis procedure, they got enough stem cells to then be edited. We got good doses of stem cells, and most of the patients engrafted by just before day 30. Just to note, that's probably about twice as long as that we would expect if you were just having an autologous bone marrow transplant for another reason. So it is slower. The cells obviously don't like being out of the body and manipulated, but again, you would probably expect that. One of the things I think is important is the platelet engraftment was pretty slow as well, and that was out at 44 days. And there was an adverse event of somebody having a bleed, a CNS bleed, which was, they survived it, but it really was quite significant. So you can get very low platelet counts during the transplant procedure. People can be quite hemorrhagic. So again, it's just underlying us thinking very carefully about this and pre-screen for any bleeding propensity or anything beforehand. But then coming on to the swimmer's plots, they've got that in orange there. That's the primary efficacy set that had enough follow-up. And again, you can see all except 3 of that set were transfusion independent and met the primary endpoint. And of the 3 that didn't, 2 subsequently did become transfusion independent on an ongoing basis, look like they're going to be transfusion independent. We don't know. This is the data we've got, such as it is, so we'd want to follow up patients for several years before we conclude that. With one patient in the washout period from having been transfused who may be becoming transfusion independent, we don't know. But again, that looks pretty encouraging as an approach. And what's really interesting is that you see these patients who may never have had a reticulocyte or haven't had a reticulocyte for many years after the procedure, starting to reproduce them very quickly and come up and have 10 grams of fetal hemoglobin. That, for me, is very impressive to see and very exciting. And coming to the point of how long this will be the case for, well, when they look at the numbers of corrected cells in the peripheral blood and in the bone marrow, then the level of correction looks stable. It doesn't look like you're losing that corrected population of stem cells, but obviously we would like to know, is that the case in 5 years' time, 10 years' time, 20 years' time, because you've got some very young patients in here. But so far, so good, I think, in terms of the correction. And then just lastly, talking about the negative side of it in terms of things like febrile neutropenia. How only 48% of them had thrombocytopenia, I don't know, but I think there must be some interesting coding of the information there. But you can't do this without becoming thrombocytopenic, really, unless they were transfused very aggressively or neutropenic. But the things that we'd expect is mucositis, which is what they saw, febrile neutropenia, the occasional cases of occlusive disease. Those are the things that we know. Until we have a more elegant way of conditioning the patients, we are going to have, that's one of the side effects of using the conditioning such as it is. No deaths, importantly. So that's a bit of a run through, and I've got another 10 minutes just to go through the sickle side of things. So again, you know this disease better than I, but again, it's thinking about what do we need to look at to take these patients into intensive therapy. So we need to know, have people had strokes? Are they at risk of bleeding in the procedure? If we're going to make people very thrombocytopenic, are livers, are hearts going to withstand, even if we are trying to dial down the conditioning that we give people, are they going to be able to tolerate that? And so it's an important thing to think about. Going back in time and looking at the outcomes, again, it started with children and having matched sibling transplants using full intensive myeloablative conditioning. And again, And sorry, this is just showing it's 1,000 patients, predominantly children, about 850 compared to 150 adults. Most of these patients are having, as I said, myeloablative conditioning from a matched sibling donor. In the adults, slightly less having full intensity transplants, some of them having less conditioning, hopefully giving them less side effects because they're older and likely to have more comorbidities. But the outcomes are good in terms of overall survival. Are very good if particularly cord or bone marrow is used as a stem cell source, and low levels of GVHD. But this is why this is in here. Again, as soon as you get to the older than 16, you get into the teenage years, patients are starting to do less well. And this is a retrospective series again, so is that because they had worse disease at the time of transplant? But there's a suspicion that as you get older, you've got more comorbidities, and so therefore the conditioning needs to be dialed down to try and prevent people running into problems. And so currently, that's why we've looked at this approach, which is being published by NIH in the States, and there are a number of series, the biggest of which was published in British Journal of Haematology, and this is bone marrow transplant dialed down about as much as you possibly can. Okay, so I'm not being blasé about it, but there's no chemotherapy in it, and there's 3 grades of total body irradiation. If you're having a transplant for, say, ALL, then you'd have 13 or 14 grades of total body irradiation. So it's not nothing, but it's much less. And the patients that we've seen going through within the national MDT who have had these sorts of transplants have tolerated it very well. Okay, but there's always a catch. There's always a negative of dialing down things too much, and that's about engraftment and the stability of the graft, and I'll come back to that. But this series had 122 patients in from centres in the US and 1 in Saudi. There was relatively standard indications of those with severe sickle phenotype, including around a fifth who'd had a stroke, and also those with evidence of cerebral vasculopathy. Engraftment happened in around day 8 to 39. But the important thing is because that conditioning is dialed down, 13%, 16 patients experienced graft failure. And that's, if you dial things down, you're going to have their old marrow, their old immune system is going to be able to reject those cells. And so that is the balance, and that's something that we bear in mind. The first 10 patients in the UK have had transplant using this approach. We are pulling apart the data of, do we need to change anything? We think preconditioning is probably an important thing, making the bone marrow quiescent, so hydroxycarbamide, exchange transfusions, sometimes top-up transfusions to make the marrow as a hospitable environment for those incoming cells. We think that's probably important, extrapolating from data in other studies and some work that's been done by VanEur in the Netherlands. I think our conclusion from the first 10 patients will be hydroxycarbamide and exchange transfusions together, please, if we can, coming in. They've used azathioprine as well, in addition, in the Netherlands, but I think we'll go with those 2 first. The amazing thing about this protocol is that of those 122 patients, 2 had graft versus host disease, which resolved with topical treatment. Now, I would never expect as a transplanter to quote that sort of rate of GVHD, and I don't when I see the patients. As I normally say, it's normally about 20-30% if you're super lucky. But it does seem to be vanishingly rare, and the thought is that actually, because you do have a degree of mixed chimerism, that you have dialed down the conditioning treatment, that's one of the reasons why you're seeing less GVHD. And these are all from matched siblings, so you'll probably only find a matched sibling that can go ahead in around somewhere 10-15% of patients, so that's a big cohort of people that we're leaving behind if they do need curative therapy. The other thing is that there's definitely a safety signal in those that had graft failure, and we saw MDS in those patients. And I think there's a bigger story, particularly from a sickle point of view. I think if you have graft failure, if you have too high degree of mixed chimerism, then you have got cells that have been stressed in a sickle bone marrow from maybe a large part of someone's life. They've then been exposed to chemotherapy and or radiotherapy, and then they're allowed to stay behind. I think that's not a good environment. I think also then if you reject your graft and you go back to having sickle, where again that marrow is particularly stressed, I think that is the combination that we want to try and eliminate from the patients and have protocols to eliminate that. And that's why, although we want people to engraft and we want them to have stable graft, I think it's even more important to push for that for those reasons. But the science of that is young, and I can't tell you that's definitely why, but that's what I'm suspicious about. There were some deaths in this group. There were 7 deaths, 5 after graft failure. So again, people doing badly, but that's those people with severe phenotype who then went back to having sickle again. That is going to be a risk. And then, as I said, the malignancy thing and side of things is something that we need to bear in mind and counsel patients very clearly too. So we decided to work together as a consortium of all of those that are interested across the country. Everyone's welcome to join, and we have monthly cellular therapy meetings with all the different centres that are interested, and we have a combined protocol. We meet, we talk about these problems, and if there are any changes, it's something that we want to do together so that we're learning as quickly as we can, and also interacting with international colleagues to learn as quickly as we can and make sure we're trying to choose the optimal protocols to use in this setting. So what happens if you don't have a matched sibling donor? Well, I will speed up. This is a retrospective study of alternate donors. So out in the front, you can see great event-free survival, I think, and overall survival in blue. But you can see those with matched unrelated donors, mismatched unrelated donors, and haploidentical or half-matched family donors, all bunching together in terms of event-free survival and their overall survival. And so from this data, we don't know what the next best option is. Often it's said that if you have a matched unrelated donor, the risk of GVHD is too high and you shouldn't use them in the sickle world. I think the studies that decision is being made of were flawed in how they delivered the graft versus host disease prophylaxis, so I don't think you can say that. But what I would concede is you're very unlikely to find a matched unrelated donor, so haploidentical transplants probably have an important role to play. And so Professor De La Fuente at St Mary's from the paediatric side, and also the Vanderbilt group have done a lot of great work trying to put together a platform that works for haploidentical transplants. This is such it is, and I don't think you'll probably be able to read that from the back, and I don't know how interested you'll be in what the exact conditioning is, but this is a lot more conditioning. It's not a myeloablative transplant per se, but it's a lot more conditioning than the matched sibling donors get, and it has TBI in it, and it has chemotherapy in it, and it also has a clever approach of giving some chemotherapy after you get the graft to try and prevent graft versus host disease, but perhaps that's something for another day. There's 2 ASH abstracts off the back of this this protocol, one in 22 and a follow-up in 23 of the 1507 study. And again, I think nearly 40-50 patients essentially did very well. Low numbers of graft failures, which is one of the things we worry about when you have a half matched, because it makes it even harder to get the graft in. So we've got 2 primary graft failures and 1 secondary graft failure there. There were 2 deaths in the cohort of 42 patients, but the rates of GVHD using this approach, which again, if you've got a half-matched donor, is something we worry about, they were acceptably low. And so if this data was put forward for any other indication other than sickle cell, I think we would say this is good enough. We're never going to get thousands of patients in a randomized controlled study. Extrapolating the use of haploidentical transplant from other settings and seeing this data, you would accept that this is looking promising for those with severe enough phenotype. So I think we are on our way to answering the question, and I think if a haploidentical transplant is probably a reasonable thing to use if you have got severe enough disease. And then that's obviously an important question to ask within your MDTs. But we do have a study open called a redress study, which is looking at haploidentical transplant in the UK, trying to recruit just over 120 patients, randomised 1-to-1 to best supportive care versus a haploidentical transplant in those 18 and above. You can have a transplant with any donor when you're less than 18 or 19, your 19th birthday, but in adults, we don't have access to haploidentical transplant outside of the redress study. Patients don't like very much that it's randomised. If you want a transplant, you want a transplant, but this is what we had to do in terms of the build, and this is the highest bar, a randomised controlled study, but it's not really been delivered in any other sphere of transplant, and it is a very high bar. But this is what we have currently. I think it's asking really good questions. It's using that Vanderbilt protocol, and which we think is looking very encouraging, and it's going to give us health economics data, it's going to give us quality of life data to prove that haploidentical is a good a choice, I believe, but we need that data. Yeah.
Well, let's see. So there was a big debate after the all-party parliamentary group
Oh, is it?
on
Okay.
cellular therapy. Because of the excellent data there is for haplo transplants, and as you say, basically it's only sickle cell that's had to prove the need in this way. There's now a debate going on in NHS England. I'll bring you into.
No, that would be good.
I somehow have ended up in these discussions.
I had somewhat of a family emergency, so I didn't make it. So I'm sad that I didn't make it. But I've said what I believe already, that I think the data would be good enough already. I think though, how long policy developments take.
Well, this was the other key thing, because we've all been through that with just access to eculizumab took 1,000 years.
Yeah.
Access to stem cell transplant took 1,000 years for adults. So unless, I think this is a shout out to policymakers, and what I'd say is, as people involved in this area. This is one of the reasons why I love being involved in this area, is that, you have to really push the policymakers, NHS England, MPs, whoever it is, to move this forward, because otherwise it's like the mills of God.
Yeah, I think we should, I think completely agree. We should do both. I think. Yeah.
Exactly. Yeah.
So that's what's happening from no, not at all. That's very helpful. Thank you. So that's what we're doing. Haploidentical point of view, and then I'm just going to round off with just going through one, coming back to lentiglobin. So that's the gene therapy approach using lentiviral vectors to introduce that anti-sickling hemoglobin gene. And there's been a number of studies that they've published from this point of view, which again show good promise of this approach in predominantly teenagers and adults. So again, 12 up. The regulatory authorities seem to like the 12 age range plus, which if someone looks after 12 plus is fine by me, but we do need to ask these questions in younger patients as well. So 35 patients here. There are a mixture with some having had stroke, but a small number have had stroke, so 40% or 5 out of those 35. But really what they showed is that they showed good production of hemoglobin A. It obviously doesn't switch off your S production, so that's why it's 50-50, because there's going to be competition between your S and also your transgene beta globin gene. And so it's going to come out at 50-50. I think that's the expected result for me. But what they did see is that they showed that the majority of the 29 that could be evaluated that had enough follow-up at this publication, it reversed their phenotype. They weren't sickling. And I think the one that did sickle it was in the context of having had an infection. There was one death, importantly, but that was a person who'd already had significant cardiac complications, and PM, they felt that it was related to cardiac fibrosis. But really importantly, and the thing that people probably have heard is the worry about malignancy after this approach. And out of a relatively low number of patients, they've had 2 patients in the first cohort, Group A, who've had MDS or acute myeloid leukemia, relatively few years after having had the gene therapy procedure. And also there's 2 patients that have anemia with trisomy 8, which are not classed as MDS, but that, if you've got a significant population of cells with trisomy 8, it's not conceptually very comfortable. We don't know why. When they went and looked back at the patients that developed MDS AML, it wasn't because of what we call insertional mutagenesis, which is when the transgene, the new gene that you're introducing, gets put into the wrong place in the genome, maybe in front of an oncogene that it then switches on. So they don't think it was a clone that had been driven by the procedure, by the gene therapy, but it is probably the fact that you've had somebody receive busulfan who already had a risk for getting a myeloid malignancy already. And so I think that's something that needs watching very carefully. it's not something that we have seen in the Vertex and the gene editing approach, but I think it's something that when we're counselling patients as well, we need to keep sort of very readily at the forefront of our mind, and scientifically this needs further pulling apart. From the Vertex, from the Exocell or Casgevy approach, this is again the ASH data from last year, where they've got 44 patients and 30 in that primary efficacy set for analysis that have enough follow-up. These patients needed more apheresis procedures, possibly because they've got less of reserve from a marrow point of view, possibly because you're using plerixafor, so harder to mobilise. But in grafting at around the same time as the transfusion-dependent thalassemia patients, a little bit earlier from a platelet point of view. But importantly, in those patients, the vast majority of them again had their phenotype reversed, and so you can see the swimmer's plot this time. The green is how far they've gone out post the procedure without having a veno-occlusive crisis. There was one patient that did have a veno-occlusive crisis, but that was in the context of them having a parvovirus infection, which probably does make some sense, and it is recovered and is now free of further events. So again, looking encouraging from this approach. This is the engraftment, the gene correction data, both in the bone marrow at the top and the peripheral blood in the lower graph, looking like the corrected cells are stable in terms of their number, and out to, not majority are out to 1 year, but we've got patients out 2 years and beyond that are looking good. Again, the side effects from the treatment is consistent with having busulfan as your conditioning agent, and that's something that we would like as a next step to stop happening, but that's next on our list. So in summary, if you're a teenager, you can have a sibling transplant. If they're matched, you can have a haploidentical or half-matched family donor. You can have an unrelated bone marrow transplant. There's no access for adolescents or adults from a lentiviral point of view from the bluebird approach, just in North America. The Exocell Casgevy gene editing approach, we're about to find out. In younger children, there are studies open. I suspect they're fully recruited. For adults, if you've got a matched sibling donor now for the last couple of years, you can. Haploidentical is the redress trial for the moment. Unrelated, just not on the cards at all. And the same answer from the bluebird point of view, and the same answer from the possibility of Exocell and Casgevy when we have a decision, maybe in the next month or so. So that's the summary. Sorry to overrun a bit. Slide.
Great.
End slideshow.
Yeah.
Any questions?
Any questions in the room? James?
Sorry, 2 questions unrelated. So, for the condition, how pre-transplant how aggressively do you isolate people? And then the second question is with the redress.
Yeah.
So you're already being conditioned in further transplant before.
So first, shall I do first?
Yeah.
First? So in terms of collation, as much as possible. And so, I mean, it's quite a long run-in time, and so from collection of the stem cells to going into a transplant, they reckon about 6 months. But even well before that, we want people to be optimally collated. And so even the patients we've had that come in on study, some of them have developed complications that you would associate with having had too much iron around. And so I'm interested what happens when you give people chemotherapy, what you then, in terms of iron, you're freeing up. Yeah. And so I think as much as possible. I mean, we've got cut-offs, but they can't be collated enough would be the answer. Yeah. And then in terms of the haploidentical transplants, and so that would be their first transplant, and so the conditioning is multi-agent chemo and a dose of TBI just at the time. Yeah.
2 questions from me. So the first one is, with the patients who are iron overloaded, do you prophylactically give them ursodeoxycholic acid?
Yes. So they get urso for sure. Everyone gets urso. We, after, try not to be anecdotal, but after our first experience, we then gave everyone defibrotide prophylaxis. But we won't get funded. If we're lucky enough that this is funded, that will not happen. And so it will be ERSO with very careful monitoring for evidence of VOD and very prompt treatment if it occurs.
Okay. And my next question, I've actually forgotten now.
No worries.
There's so many questions, but I'll come back.
One last question from Emma.
So sort of related. So do you think there's a role for given the potential role of non-transferable bound iron? Do you think there's a role for giving desferal instead of defibrotase? Because it's a lot cheaper.
Well, it would be. I don't see why not. And I think. And I also think what's going on at the time. So as people come in, we stop their collation. We're worried about them in grafting, and so all the collation stops during the transplant procedure and possibly actually at the time of collection as well. Just we think if we can have a window, then that's a sensible thing to do. But I wonder what else we could do at the time of transplant. And so there are, if patients from a malignant point of view are at risk of cardiomyopathy, then obviously there are collating agents that are given as they get drugs like anthracyclines. And is that what's going on? Should we actually be braver and be mopping up iron even when going into the transplant? I mean, you'll be able to tell me about the PK of all these things, but is it that much of a concern if we then cover them during the actual time they've been conditioned and then we have a washout and then we give them their cells? I don't know.
Yeah, no, I think it would be interesting.
Yeah.
Yeah. Great. Thank you very much, Ben. Super. So now we've got Dr. Vikram Talurikar, who is our reproductive medicine specialist at UCH, and he's going to talk about the ovarian reserve and menopause in haemoglobinopathies.